So I will be talking about automated organoid assays using the microfluidic-based Pu.MA system. So today I will talk about Pu.MA system and how it works. I'll walk you on how to use organoid assay workflows with Pu.MA system. And I'll show you an example of the data and assays that we use to do multifunctional profiling of 3D cell model. Now, our vision is to have easy-to-use open lab on a chip for doing 3D assays where you can maintain physiological conditions. The system that would accommodate different types of organoids. They could range in size between 100 microns to 1 millimeter. If needed, you can use some matrix for these assays. We automated all complex assay steps for you, which really allows spend less time, less reagents on running complex assays. It allows to minimize handling errors and get really high quality and reproducible data with your precious organoid samples. Now here I mapped out a workflow that we developed and use with the system. The first step would be to load your sample, organoid or spheroid, in the Flowchip. Then the chip is placed inside the machine, and machine itself is sitting inside the cell culture incubator. So essentially you run all your assays under standard cell culture conditions. Then you have to select the protocol, which is preloaded in the machine that you would like to run. Protocol is completed, you take your plate out, and you can use it for microscopy. It is possible to do fluorescence microscopy. You can do also luminescence-based readout. You can do plate reader-based downstream assays. And if needed, we can collect samples also for downstream analysis, for example, for metabolites assessments. Now, the heart of the system is the Flowchip. Here you can see the frame with 4 Flowchips in it, and you can use in your assays between 1 and 4, depending on the needs. Each Flowchip has 8 test lanes, and each lane is designated to 1 sample. So essentially, you can do between 8 and 32 samples. Again, depends on how would you like to do your assay. Now, in each lane, there's a sample well where the organoid or spheroid is positioned. And the sample well is connected to a number of reservoir wells through microfluidic channels. And these reservoir wells, you can fill up with anything, like, for example, medium with compounds. It could be different washing buffers. It could be staining buffers. They can be used for collecting supernatants. So highly customizable depending on the configuration of the assay that you would like to run. Now, the well itself is pretty small. The volume of the well is 20 microliters, and you can see here that your sample is positioned at the very bottom in the protected sample chamber, what we call it. The diameter of the protected sample is about 1. It is optically suitable for doing high-resolution imaging. And overall, the dimensions of this frame and the Flowchip resembles standard 384 well plate with just minor modifications. So this plate is absolutely suitable and compatible with all instrumentation like standard plate readers or microscope or high-content imagers. Now, like I said, the protected sample chamber is our key proprietary technology. So here you can see how the sample organoid is loaded into the well. And again, it is positioned inside this protected chamber. We can do fluid exchange from the channels, and we can do up to 95% exchange. The volume of this protected chamber is about 1 microliter. Now, if there's a requirement to, for example, drain your well completely and remove medium completely, you can do so by doing it from the bottom of the chip. Now, when the Flowchip is loaded, sample is there, and all reagents are loaded, you are ready to start your assay. The interface is really simple to use. You just have to select the protocol, and the list of protocols is already preloaded on the machine. And then you click the run button and kind of walk away and let the machine do all the steps of the assay for you. Let's take a closer look what's happening inside the Flowchip while the assay is running. So here you can see an example, an outline of the Flowchip with the sample. First thing that we can do, for example, we can remove old medium and replace it with fresh medium. Let the sample incubate for desired amount of time. Then we can remove this medium and transfer compounds. Let the sample be treated with the compounds. Then we can collect supernatant back in this well and transfer staining solution. Let our sample stain, remove stains, and do a wash step if needed. Now, at this point, when all the transfers and sample collection is completed, your assay is done. and the plate is ready to use for the downstream analysis. You can really use different kind of samples with the machine. It could be single cell spheroids formed from standard cell lines. It could be tumoroids developed from primary tumors. We also did assays with primary and iPSC-derived organoids. Organoids typically require matrix for culture, and you can use matrigel with the system. So here, I would like to show you an example of how we do multifunctional profiling. This study was done using breast cancer patient-derived tumoroids that were loaded in the plate and treated with different compounds. After that, we did viability assessment with staining for live dead these tumoroids and did confocal imaging. Also, from the same samples, we collected supernatant and later did downstream analysis of metabolites. And also at the end, we did immunofluorescence staining for biomarkers and again did confocal imaging for that. So you can really see here multi-step complex assays that could be used with the machine automatically and allows you to collect a lot of data to do multifunctional profiling of your samples. So to conclude my short introduction of the technology, I would like to share with you this long organoid image that we obtained by staining and using staining protocol with our Pu.MA system and confocal imaging. And I also would like to mention here that very soon we will release our new technical note on how to do assays with matrigel with our Flowchip. And also in July, we will do another webinar on the same topic, going into details and aspects of using matrigel. So please keep an eye on our announcement and register. So if you have any questions or would like to discuss your particular assays and how we can use Pu.MA system for that, please reach out at any time, and I would be happy to answer your questions.